Hey everyone, here we are. I'm going to talk a little bit more about genetics, and in this lecture we're going to start talking about DNA replication. So this is really important stuff no matter what field of biology you're working in. So let's get started. So in this first part we're going to talk about the structure of DNA. We've talked about nucleic acids a little bit. We're going to go into a little bit more detail. And then in the next part, we're going to talk about how DNA gets replicated and how do mutations occur, what happens with mutations. And so why do you need to know this? Well, I mean, this is the discovery in biology. It's a fundamental concept no matter what you're studying. It's just so important to living things. Understanding and figuring out the structure of DNA in the 20th century was a huge deal. And now if you want to be a 21st century biologist, you have to really understand your DNA. And so, again, a lot of us, you might be like me, I'm an organismal guy, big picture guy. And so you might ask, why do I need to understand this? I'm getting tired of all this cellular, molecular stuff. But really, this is where it happens. And by understanding how the DNA works, that explains so much in biology. And so if you want to be, a biologist, you need to have a good understanding of this. And of course, most of you will take genetics, where you'll get into even more detail. So it's, it's just one of those things that's really important to biology. So let's start talking about the structure of DNA a little bit. So the first question is DNA or protein? Okay, so you know that DNA is the source of inheritance, right? That the genes get passed on through the DNA. You know that. You know that DNA makes RNA, makes protein. We've said this 100 times. And you know that a gene is a piece of DNA or a piece of a chromosome that codes for a protein. And you know that Mendelian inheritance and the laws of genetic inheritance that Mendel came up with can be explained by understanding chromosomes and their movement during meiosis. But of course, we didn't always know all of this. And so in the early 20th century, in the early 1900s, scientists were not sure what was the molecule of inheritance. They knew that there was nucleic acids and they knew there was proteins, but was it DNA or was it protein that passed along the traits from one generation to the other? And so that was kind of the first big step in the early 20th century. Once it was pretty well determined that, okay, it's DNA that is the molecule that passes on traits, the molecule of inheritance, then it became important to understand the structure. And once you understand the structure, then you can really understand how it passes those traits on. And so Watson and Crick are the famous names associated with describing the double helix and the structure of DNA. And this would have been in the mid-20th century, mid-1900s. But we always like to point out that a name that you don't often hear, Watson and Crick is kind of a famous name when you talk about DNA. A name that's also should be as famous is Rosalind Franklin. She rarely gets any credit, but she was an expert in X-ray crystallography. And it was using her X-ray crystallographs, I guess you call them, I don't know what you call them, her pictures from her X-ray crystallography, that's what allowed them to figure out the structure of the DNA. So Rosalind Franklin also should get a lot of credit for this. And so this is what one of those micrographs looks like. And so basically you're shooting X-rays and things at the molecule and then developing film. And by the way that the rays get bent and the way the film develops, you can infer what the structure of the molecule is. So this is what Watson and Crick based their model on. Now let's go back and review nucleic acids a little bit. Remember that nucleic acids are made from nucleotides. That's the monomer. And it's just a long chain of them, right? There aren't any branches. It's just a long chain of nucleotides holding hands. Those nucleotides have three parts. They have a nitrogenous base, a pentose sugar, a 5-carbon sugar, and a phosphate group. And so here's a strand of DNA and it's illustrating one of these nucleotides that's part of the strand. And I'm going to back up there for a second. Again, if you zoom in on one of those nucleotides, you can see the nitrogenous base is attached to the pentose sugar, and then there's also a phosphate. That's one nucleotide. And you recall that there's only 5 of those nitrogenous bases: cytosine, thymine, uracil, adenine, and guanine. You recall that thymine is the is used in DNA, and uracil is the one that's used in RNA. And the other 3 are used in both. And of those pentose sugars, there's only 2 pentose sugars you're going to find: ribose and deoxyribose. And so you also remember, we talked about this earlier, the differences between DNA and RNA. DNA is double-stranded, it has 2 strands. And because it has 2 strands, and they're both long strings, that allows those nitrogenous bases to pair up. And they don't just pair up at random, they only pair up in a particular way. And so A always pairs with T, and C always pairs with G. And so the way you can remember this is all tigers can growl. A's with T's, C's with G's. And so you never have like a C with a C, or you never have a C with an A. It's always an A with a T and a C with a G. But we'll find out that this fact is what makes all the rest of the stuff we're going to learn possible. And that's because if you have one strand of DNA, you automatically know the other strand that goes with it, because you can only pair these bases up in a particular way. And so, for example, here's one strand of DNA. And so what's the other strand? Well, I know it. I know that A's are with T's and C's are with G's. And so I can create what's known as the complementary strand. And so if you know one, if you have one strand, there's always a complement to it in DNA. And so we can practice this, and I'll ask you this question a lot. Let's say you've got a piece of DNA, AA, CTA, GGG. Which of these would be the complementary strand? Well, you could start, A's must pair with T's, so starting on the left, A's must pair with T's, so the complementary strand must start with a TT. So we can rule out choice A, we can rule out choice C. The next nucleotide in our original strand is C, so in the complementary strand, the third nucleotide must be a G, and so you see that rules out choice B, and so then you're left with choice D. And so, again, by knowing all tigers can growl, you can always figure out the complementary strand. So you always have these 2 strands, and you've got the nitrogenous bases in the middle. Imagine a ladder, and if you take the ladder and you grab it at the top and bottom and then you twist it, that's a double helix. And that's the overall structure of DNA. And so again, if you look at this figure, you can see how the bases are pairing up, and there's always an A is always with a T and a C is always with a G. You can see that the bases sort of make the rungs of the ladder. So if you think about a ladder, the part that you crawl on, you step on, and you grab hold of, those are the rungs. And that, the metaphor is those are the bases. And then the sugar phosphate backbone makes the sides of the ladder. And so again, just another look at it. It's a figure from your book. And you see the sugar phosphate backbone. So it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. and then the nitrogenous bases are all stuck to one side. And so this would be one strand, and then we could easily build the complementary strand based on what we know, based upon A's pairing with T's and C's pairing with G's. And so this is just another look at the exact same thing again. And you can see sugar, the backbone, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then on each of those sugars, there's a nitrogenous base sticking out. And they're all sticking out to one side. Now this is one strand. If this is DNA, we need another strand, right? So let's zoom in again on the nucleotide, and specifically on the sugar phosphate of one of these nucleotides in the strand. And so something that you gotta, you're just gonna hear these terms so much, you really need to understand exactly what we're talking about. Look at how the carbons are numbered. So in organic chemistry, we've talked about this already. You know, you've got these rings and you've got a number of carbons, and there are certain ways that you number them. So you can always, you know, scientists can talk about the carbons, and we all know what everyone else is talking about. And so look at how these carbons are numbered. They're numbered in clockwise fashion, starting with 1. It's actually 1 prime, so that little apostrophe with the number, they call that the prime. So you got 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And they're always numbered in this manner. And this way we can talk about what's going on at each carbon. And so for example, you notice that the 5 prime carbon is actually sticking up. Remember, you know, these have a 3-dimensional structure. And so these rings are like in a plane. And so carbons 1 through 4 are in a plane, but you see there's also an oxygen there to make the pentose. And that 5th carbon is sort of sticking up from the plane. And so by numbering these, we can talk about what's going on at each carbon. And so the nitrogenous base is always bound to the 1 prime carbon, or the number 1 carbon. And that's how then they can always be stuck off to one side, because they're always stuck on the same carbon. When we're joining 2 nucleotides together, we join them, the new nucleotide joins at the 3 prime carbon. And so when that new nucleotide comes in, it's going to attach to the 3 prime carbon, and then the phosphate that goes with this sugar is attached to the 5 prime carbon. And so that's going to be real important when we talk about building a DNA structure later. Because these are arranged in this way, and there's always, the nitrogenous base is always on the 1 prime carbon, the phosphate's always on the 5 prime carbon. When you stick these nucleotides together, it gives the entire strand directionality. And we talk about the 5 prime end and the 3 prime end. So you look at these, look at the nucleotides here. They're not just stuck together, they're not just sort of rammed together in any order. They're in a very particular order. And so you've got the first nucleotide at the top here. You can see the very tippy top of this strand is where the 5 prime carbon is for that nucleotide. And then the next nucleotide is attached to its 3 prime carbon, and then so on and so on. And so you go all the way down to the bottom of the strand that you're looking at, and you can see that the open carbon at the very end is a 3 prime carbon, and it's always that way. And so the end of that one end is always going to be a 5 prime carbon of one nucleotide, and the other end of the strand is going to be a 3 prime carbon of a different nucleotide. But that 3 prime carbon is always open. So there's always an opening at one end of the strand, but not the other end. That's going to be real important when we're trying to build this strand, build DNA later. But this is a term you're going to hear all the time. 5 prime end, 3 prime end. Well, this is what it's based on. It's based upon the numbering of those carbons. So here's that figure from your book. And again, you can see they've labeled the 5' end and the 3' end. And if you look at those carbons in the pentose sugar, you can see where that 5' and 3' comes in. And you can see that down on that 3' end, you've got an open carbon. The 3' carbon of the very last nucleotide doesn't have a phosphate, doesn't have anything attached to it. That's going to be important. And so here's just another drawing of the same thing, just trying to, again, draw all these ideas together. Now, one thing that you should notice from this picture, now we have the 2 strands of DNA. Earlier we were just showing 1 strand, but DNA is 2-stranded. And so here we see both strands of DNA, but notice the orientation of each strand's 5' end and each strand's 3' end. You see they run in opposite directions, and we call this anti-parallel. So you see how one strand goes from 5' at the top of the slide to 3' at the bottom. Its complement has the 5' end at the bottom and the 3' end at the top. And so they both have that directionality, but that directionality goes in opposite directions. That's anti-parallel. And so now when those 2 strands come together, how are they held together? They're held together by hydrogen bonds between those nitrogenous bases. And so you recall, hydrogen bonds are the weakest kind of bonds out there, and hydrogen bonds are usually between different molecules, not within a molecule. They're not nearly as strong as a covalent bond. But if you have a lot of them, they can be strong. But they're not as strong as covalent bonds. And so here we're showing an adenine pairing with a thymine at top, and a guanine and a cytosine bonding at the bottom. And you can see with the little dotted lines, they're representing the hydrogen bonds that occur between these bases. And so you can see, the G to C bond takes 3 hydrogen bonds, or when G and C bond together, it's 3 hydrogen bonds, and when A and T bond together, it's only 2 hydrogen bonds. But it's hydrogen bonds. So, a lot of them makes it strong, but they're also easy to pull apart if you need to pull this apart. And so again, another figure from your book showing the 2 strands. You see they're anti-parallel. Look at the 5' end and the 3' end of each strand, and you see how they're running in different directions. You see that A's are always with T's, C's are always with G's. And you can see the hydrogen bonds represented between those nitrogenous bases. And this is what I just said earlier. A to T is held together with 2 hydrogen bonds. C to G is held together with 3 hydrogen bonds. So C and G are held together just a smidge more tightly than A to T, because you've got an extra hydrogen bond there. Note, if you look at these nitrogenous bases, a couple of them are double ringed and a couple of them are single ringed. You see that? You see the adenine and guanine have 2 rings, thymine and cytosine have only 1 ring. But a single ring always pairs with a double ring. You never get 2 double rings together or 2 single rings together. And so that's important because it keeps the DNA the same width, right? If you had 2 double rings together, those are bigger molecules, and so the DNA would bulge out. And if you had 2 single rings that paired together, the DNA would come way in. But by always having C with G and A with T, it's always a double ring with a single ring, so it's always the exact same width all the way down, and that's more energetically stable. So here's another figure from your book, again just emphasizing those hydrogen bonds between the nitrogenous bases. Okay, so that's a little bit more detail on the structure of DNA, but I purposely emphasized some of those points because in the next section, in the next lecture, we're going to start building a new DNA. We're going to replicate DNA, and so those details I gave you are going to be important, and they're going to help explain a lot of the details of building the new DNA. So that'll be in the next lecture. So let me know if you've got any questions, and I'll see you next time I see you. We're going to build some DNA. Awesome. All right, see you in a bit.